All right, so I'd like to talk to you about some of the work that we're doing in my lab, synthesizing anti-malarial and anti-tumor agents using the Yugi reaction. So first of all, why are we working on malaria? Well, it turns out that it's still a really huge problem. I don't know if everybody realizes it, but there are at least 300 million people a year infected and over 1 million deaths per year. These are pretty conservative numbers, actually. We don't see that in the United States, but around the world, it really is still a huge problem. And there are drugs, in fact, that cure malaria, but unless you actually eradicate a disease, you're always fighting because you're going to develop resistance. So there's always a need to develop new compounds that attack the disease in new ways. And the strategy that we're using is the Yugi reaction. So this is actually a pretty old reaction, and it's been very useful over the years because what you're doing is you're taking 4 pools of common compounds that you can make, fairly cheaply. You have an aldehyde or a ketone, an amine, an isonitrile, and a carboxylic acid, and you mix them together in methanol at room temperature, and 24 hours later, if you're lucky, this will actually precipitate out of the solution. So it's extremely easy to do, and it has a huge potential for making very large libraries very quickly. So you can make a huge diversity of compounds pretty easily. And in terms of whether we isolate these compounds, how we isolate them, I'm particularly interested in the compounds that actually precipitate. They don't always do that, but when they do, you can imagine how useful it could be to have a library of compounds that only precipitate. So we do have a project right now that are trying to understand what substituents actually lead to that kind of behavior to make it easy to isolate. So what we've been doing is using a virtual library of 500,000 compounds. So what that means is we basically have a web service that processes pools of, maybe 20 to 30 of each of these compounds, and then it generates very large lists of the combinations. So when I refer to the virtual library, that's what I mean, is the list of those. Now, it turns out that the Yugi products actually do bind to some malarial enzymes. So if we take that large library and we start to do docking, we find that, for example, with enoyl reductase, which is an enzyme that the malarial parasite needs to build its cell wall, that you can in fact fit it into the active site of that enzyme. And you can see pictures here of sometimes you have 2 different isomers and they both dock, but they dock in slightly different ways. Now, recently, we've been very interested in falcipain. This is an enzyme, actually, that the malarial parasite uses to break down hemoglobin. And it was discovered at the Rosenthal group, University of California, San Francisco. And they've been actually kind enough to collaborate with us to test our compounds. So I'm very excited by this because we have actually a way of shipping compounds over there that we make in the lab and finding out if our hypothesis is correct, the way that we've been, trying to make them. So one of the things I need to mention about how we do research in my lab is we do it under open conditions. So that means basically that on a day-to-day basis, what happens in the lab and our discussions and things like that are communicated to the world. And we use blogs, wikis, even Second Life, and mailing lists to communicate that information. And what that's actually permitted, it's enabled us to have collaborations with people that we would have never met any other way. For example, Rajarshi Guha at Indiana University, we met through our blogs, and he's actually been very active in docking our compounds to tell us which compounds we should make for enoyl reductase or for falcipain, for example. We have another collaborator in Singapore, Tsu 10. And we have the Rosenthal group, which is willing to test our compounds. And again, it's very unexpected the things that happen when you start to do research in the open. I was contacted by Dan Zaharovitz at National Cancer Institute, and he says, hey, why don't you send us your compounds and we can test them. And they've in fact been testing a few of our compounds. And it just so happens, small world, Rajarshi actually had written an algorithm to predict anti-tumor activity for compounds. So everything connects together, and so we're able to take these large libraries and ask the questions, which one of these molecules should we make? And then we actually have the ability to test them. So this is very nice from an open science standpoint because we have a complete loop. So in other words, we have a hypothesis, we can select the compounds. At Drexel, we can make them. Elsewhere, either at NCI or in San Francisco, wherever people are willing to test them, we can do that. And then we can get feedback as to whether or not we're taking the right approach. And we're doing all of this in the open, which is very exciting. This idea of doing science in the open, of talking about your research results prior to a full publication in a journal, has actually captured the imagination of a lot of people recently. UsefulChem, which is the title of the project, this Malaria project, also referred to as Open Notebook Science, which is a term that I coined to refer to doing science where your actual laboratory notebook is completely public. Just this week, in fact, Wired Magazine has talked about what we're doing. We've had multiple citations in Nature blogs. CNE News has ran a couple of articles, a big one last summer. Cell has discussed it, per contra, assignment 0, NPR Marketplace, Chemistry Central, 3 Quarks Daily. So basically, there's definitely a movement afoot, and I think that we really can't ignore this. And so what I'm trying to show here are the useful things that we can do when we do go open. So just to give you a few screenshots of what does this mean when I talk about our lab book is open. Well, here's what it actually looks like. We keep our experiments on a wiki. So each page of a wiki is a separate experiment. And a wiki, you see this little button here that says edit this page. It's very easy to edit it. So there's not much, technological training that you need to do for anybody that's working here. The nice thing about the wiki is you can track the edits. So if I make a change, I can then compare 2 versions, and the stuff in red will be the stuff that was deleted, the stuff in green will be added. And so you can, over time, you can actually see every single edit. So if you have a student that's doing an experiment in the lab, you can see exactly the order in which everything was done. You can see the discussion points. You can see error correction. All of that kind of stuff is all tractable and available online, on tap, essentially. So to actually monitor what's going on in our lab, it's really as simple as clicking on the recent change button, and you can see, in the past few hours exactly what got added, including my comments as I read the experiments. We use Wikispaces, which is a hosted third-party application, and a big advantage of that is that you actually get third-party timestamps. So if it comes down to someone trying to plagiarize what you've done, or trying to claim that they had an idea before you did, because we have this large company that actually maintains a completely separate third-party timestamp, it's pretty easy to prove that you knew what when. It's certainly safer than sending in your proposal for review that only your closest competitors will review, and you really have no way of proving that what was there. So when I talk about doing things in the open, I really do mean everything, in the sense that in a traditional article, you may have a printout of a spectrum. Here in UsefulChem, we actually use JSpecView, which our NMR spectra are interactive. So if you wanted to expand a peak, for example, using your browser, you can actually blow it up and measure the J constant, for example. So that means that we're open to interpretation. So we have to stand by what we say. We have the raw data. We can link to it. And if people don't agree with our conclusions, they can certainly go in and take a look at the raw data and make their thoughts known. And so there's a wiki part. There's also a blog component. The blog component is really at a much higher level. That's where I will talk about milestones. So, if we've been trying to get something solved and then all of a sudden we have it solved, or if we have a problem that's been really difficult to wrap our minds around, we're able to actually, write that in the blog. And there are people who read that can participate. So, because I'm out of time, right? So basically, we can get feedback from the outside. We also have mailing lists to interact with our collaborators from Indiana University in Singapore. And there's some additional things if you're interested. We do some mechanistic work as well, where we're seeing the cleavage of referral groups in acid. So I don't have time now, but certainly for students interested in that, I'd be happy to discuss it. And that's it.
Thank you very much.